The overall goal of this method is to use cell tracking dyes to directly quantify the extent of cell division for different immune cell subsets within complex populations. This is achieved by first labeling the parent cell population with a fluorescent dye known to give stable binding to cellular proteins or to intercalate non-covalently into cellular membranes. For tracking dye-labeled cells that respond to stimulation, counterstaining with fluorescent antibodies, and analysis using multi-parameter flow cytometry allows detection of differential responses among the immune cell types of interest. Stimulated but tracking dye unstained controls are used to establish the lowest daughter cell fluorescence intensity that can be distinguished from autofluorescence. Tracking dye-labeled but unstimulated controls are then used to establish the fluorescence intensity at which the undivided cells will be detected. Stimulation typically results in a broad range of intensities as cells proliferating in response become progressively dimmer, but non-responders do not. Ultimately, the use of peak modeling software to estimate the relative frequency of the cells in different generations can allow quantitative comparison of the proliferative responses across different populations or stimuli, using metrics such as proliferation index or precursor frequency.
The main advantage of this technique over existing methods, like tritiated thymidine incorporation or Ki67 expression, is that the dye dilution provides a direct measure of cell division that can be combined